membrane. So you can see a microscopic figure over here, electron microscopic figure. So it is a bilipid layer. So two layers you can see of the lipid. So then it is made up of proteins, carbohydrate chains, and phospholipids and all. So there are some transport channels also through which the transportation of materials takes place in and out of the cell. So the cell membrane is very, very flexible. So moving on to the textbook page. So cell actually has the plasma membrane, nucleus and the cytoplasm as the major parts. So now moving on to the cell membrane or the plasma membrane. So this is the outermost covering of the shell and it separates the contents of the cell from its external environment. The plasma membrane allows or permits the entry and exit of selected materials in and out of the cell. So it also prevents the movement of some other materials also. So the cell membrane is also called as selectively permeable membrane. It's also called as plasma membrane, cell membrane, selectively permeable membrane or semi-permeable membrane. So how does the movement of substances takes place into the cell and out of the cell? So let us come to know. So for example, some substances like carbon dioxide or oxygen can move across the cell, means in and out of the cell, by a process called as diffusion. So in your earlier classes you have learnt about the diffusion, right? So what is it? So it is the spontaneous movement of a substance from a region of higher concentration to a region of lower concentration. So something similar to this happens in the cells also. For example, the carbon dioxide, which is a cellular waste and it requires to be excreted out of the cell. So inside the cell, the carbon dioxide, it accumulates, gathers or gets together in a high concentration inside the cell. So rather than the outside the cell, that is the external environment. So the concentration of the carbon dioxide is very, very low outside the cell than the inside the cell. So as soon as there is a difference of concentration, I repeat, there is a difference of concentration of carbon dioxide inside and outside of a cell. So the carbon dioxide moves out of the cell because it moves from a region of higher concentration to a region of lower concentration. Through what? Through the semi-permeable membrane or the plasma membrane. By which process? By the process of diffusion. Similarly, the oxygen also, it enters the cell by the process of diffusion only. So diffusion plays an important role in the gaseous exchange between the cell and its external environment. So water also obeys the law of diffusion. So how it is, let us see now. The movement of water molecules through such a selectively permeable membrane is called as osmosis. The movement of water across the plasma membrane is also affected by the amount of substance dissolved in the water. So thus, osmosis is the passage of water from a region of high water concentration through a selectively permeable membrane to a region of low water concentration till equilibrium or equality is reached. I repeat, what is osmosis? The passage of water from a region of higher water concentration through a selectively permeable membrane to a region of low water concentration till equilibrium is reached. So now let us discuss what will happen if we put an animal cell or a plant cell into a solution of sugar or salt in water. So the first condition goes like this. So the following three things can happen. What is the first thing? If the medium surrounding the cell has the high water concentration than the cell, I repeat, we take in a jar, we take some water and we put a very little content of salt or sugar. Okay, so this medium is called as the hypotonic solution. So, if the medium surrounding the cell, and we put into this beaker one cell. So, the medium surrounding the cell has high water concentration than the cell inside. So, meaning that the outside solution is very dilute. The cell will gain water by osmosis, because in the beaker the salt content is very less, salt or sugar. The water content is very high. But the cell which is inside this medium, so what happens? The water molecules moves inside the cell by the process of osmosis. Why? Because there is a difference in concentration. Okay. So, such a solution is called as hypotonic solution. I repeat, hypotonic solution. So, here water molecules are free to pass across the cell membrane. So, the water molecules moves in and out of the cell membrane in both the directions. But more water will come inside the cell. So, more water molecules will enter the cell from the hypotonic solution. So, the net result, what is the result? The water enters the cell more and the cell will lightly swell up. So, what happens? The cell regains the water and ಸ್ವಲ್ಪ ದಪ್ಪ ಆಗುತ್ತದು. Okay, it swells up. So, the second condition, what do we have? So, if the medium has exactly the same water concentration as the cell. So, in a beaker we have taken some water. So, we have water we have taken and we have added little bit of salt and sugar to that, and we have kept a cell in that. It could be a plant cell or an animal cell. So, the medium has exactly the same water concentration as the cell. So, there will be no movement of water across the cell membrane. Such a solution is known as an isotonic solution. What do we call it as? Isotonic solution. Here the water concentration is exactly the same in the cell and in the beaker or the medium. So, water crosses the cell membrane in both the directions. The water moves inside the cell through the cell membrane, and it moves outside the cell also through the cell membrane. But the amount going in and out is the same. The movement of water molecules going inside the cell and outside the cell is the same. So, the cell will stay the same size. So, what is the result? So, cell will have its same size. ಅದು ದಪ್ಪನೂ ಆಗಲ್ಲ, ಸಣ್ಣನೂ ಆಗಲ್ಲ. It remains the same size. Now, the third condition, what is happening over here? If the medium has low concentration of water, means what we should do now? In the beaker, we have taken some water. So, to that, we should add salt or sugar content more. So, the concentration will be very high and the water will be very less. So, meaning that it is a very concentrated solution. The cell will lose water by the osmosis. So, to this concentrated solution, when we put a cell, what happens? The cell will lose the water. By which process? By the osmosis. So, such a solution which we have taken in the beaker is called as hypertonic solution. The salt or the sugar content is very high than the water. Again, water crosses the cell membrane in both the directions. So, in this condition, the water moves in and out of the cell membrane. But this time what happens? The water leaves the cell more. So, from the cell, the water content will go more to the outside environment. Therefore, what is the result? The cell will shrink. The cell will shrink, lose its size. ಸಣ್ಣ ಆಗೋಗುತ್ತೆ. So, thus, osmosis is a special case of diffusion through a selectively permeable membrane. So, let us now try out an activity. So, activity 5.3, osmosis with an egg. So, what we have to do is, we have to remove the shell of the egg. How? By dissolving it in dilute hydrochloric acid. So then the shell is mostly the calcium carbonate. The white color content, what you see the shell, it is actually the calcium carbonate. So after putting this shell into a dilute hydrochloric solution, this calcium carbonate will get dissolved. So what you see, a thin outer skin is now encloses the egg. Put the egg in pure water and observe after 5 minutes. So what do you see? The egg swells, means the egg ಏನಾಗುತ್ತೆ ದಪ್ಪ ಆಗೋಗುತ್ತೆ, swell ಆಗುತ್ತೆ, because water passes into the egg by the process of osmosis. So now next condition, place the same deshelled egg in a concentrated salt solution. So now in the beaker we have to take more salt content and less of water. We have to dissolve it and we should put this deshelled egg into that. After 5 minutes, what do you see actually? The water passes out of the egg solution and into the salt solution. So the egg actually, it shrinks down. ಸಣ್ಣ ಆಗೋಗುತ್ತೆ ಇಲ್ಲಿ. Okay? So why? Because the salt is more in the beaker. Okay? So now the same thing you can try it out with the apricots or the raisins. So put dry raisins or the apricots in a plain water and leave them for some time. Ona drakshi, dry raisins andre. So then place them into a concentrated solution of salt or sugar. So what do you see next? So in the condition A, it gains water and swells up. So in the concentrated solution, what happens? It actually shrinks down. So let us see how it happens. So there are 3 types of solutions. So first one, we are talking about the hypotonic solution. It contains less solute and more water. More water is seen over here compared to the another solution. So water diffuses across the semi-permeable membrane, means water moves in and out of the cell membrane. So out of a hypotonic solution into the egg. So movement of more water molecules inside the egg happens. So because of that, what happens? The egg or the cell expands. Okay. So next it is the hypertonic solution, means more of the solutes, the salt is very high in this solution than the water. So when you put a cell or an egg, what happens? The water diffusion takes place across the semi-permeable membrane into the hypertonic solution. So the water molecules from the egg or the cell moves out of the semi-permeable membrane into the medium. So what is happening? The cell will shrink down. The water molecules ಎಲ್ಲ ಹೊರಗಡೆ ಹೋದ್ಮೇಲೆ ಏನಾಗುತ್ತೆ? ಆ cell shrink ಆಗುತ್ತೆ, ಚಿಕ್ಕದಾಗೋಗುತ್ತೆ. So the third condition is isotonic solution. So now the same concentration of solutes is taken in the beaker, the equal amount of water as it is present in the cell also. So what happens? So there is no movement. Movement happens, but it is equal. Water moves in and out of the semi-permeable membrane. So in isotonic solution, the cell remains normal. There is no change in the shape of the cell. So moving on to the next activity. So when egg is placed in the mineral acid solution. So after some time, what happens? The shell actually get dissolved. So then when you place it in hypertonic solution, means the solute content is more. Okay. So what happens? After some time, you can see the size of the egg is reduced. So osmosis has taken place. So the water molecules from the egg has moved out into the hypertonic solution. So next condition, what to do? So put this egg into the water. So which is the hypotonic solution. Hypotonic means the solute content are very less. The salt or the sugar content is very less, but the water molecules are more. So what happens? The egg regains the shape due to osmosis. The water molecules from the hypotonic solution moves inside the egg. So what happens? the egg regains its shape. ಮತ್ತೆ normal shape ಬರುತ್ತೆ egg. Okay students? So next activity. So difference between the endosmosis and the exosmosis. It's nothing but raisins. Onadrakshi idiyalva? So if you put it in water, so what happens? It swells up. So that is called as endosmosis. The inflow of the solvent water, the inflow of the water molecules into the cell from the outside. When cell is placed in distilled water, the cell actually swells up. Dappa agutte neeral hakida takshana. Exosmosis. So when you put these grapes, actually the grapes into the salt solution, tumba salt irutte ee solution alli, anta solution ge ನೀವು grapes ಹಾಕಿದ್ರೆ ಏನಾಗುತ್ತೆ? Actually in the grapes the water content will move out. So these grapes will become very very dry and shrunken. ಒಣಗೋಗುತ್ತಲ್ಲ. So the outward flow of water from the cell when placed in more concentrated solution like sugar solution means the hypertonic solution, the cell actually shrinks in this case. Okay, so this is all about endosmosis and the exosmosis. So moving on to the textbook pic, so the last part. In unicellular freshwater organisms and the most plant cells tend to gain water through the osmosis. So absorption of water by the plant roots also is an example of the osmosis. So how the plants absorb the water by the roots is nothing but through the process of osmosis. So thus diffusion is important in exchange of gases and the water in the life of a cell. In addition to this, the cell also obtains nutrition from its environment. So different molecules moves in and out of the cell through a type of transport requiring the use of energy. So in your last classes we had discussed that the different molecules in and out of the cell through a type of transport requiring energy. So energy requiring transport is called as what? Active transport. So what is the energy molecules which have been spent over here? It is nothing but the ATP. ATP molecules. What are ATP? What is the full form of ATP? Adenosine triphosphate molecules. When energy is spent for the movement of the molecules in and out, so it is called as the active transport. So when no energy like ATP is spent for the movement of the molecules in and out of the cell, so that is called as the passive transport. The plasma membrane is flexible. The plasma membrane is very very flexible and is made up of the organic molecules called as the lipids and the proteins. So however we observe the structure of the plasma membrane only through an electron microscope. So the flexibility of the cell membrane also enables the cell to engulf in food, take in the food and other materials also from its external environment. So such processes are known as the endocytosis. I repeat, the process through which the materials can be engulfed by these unicellular organisms from the external environment. So the process is called as endocytosis. So this is how amoeba, the unicellular organism, acquires its food through endocytosis. So I have a slide for you people. So this is, so amoeba, so it takes in the nutrition in the form of holozoic nutrition. So the amoeba senses the food. So and the pseudopodia actually surrounds the food. Okay, so what is the food actually of amoeba? It is the zooplanktons or the phytoplanktons. So this is called as the ingestion, taking in food by the amoeba through the pseudopodia, which is the finger-like projection which surrounds around the food molecule, is called as the ingestion. So this is the first process. So next what happens? The food is enclosed into a food vacuole. So this food vacuole which is engulfed inside the amoeba, so it has the food along with the water. So the food particle which is engulfed in the form of the food vacuole. So this process is called as the digestion. Okay, so next what happens? So the engulfed food is actually some enzymes from the cytoplasm are secreted into the food vacuole so that the digestion process completes. Okay, so because it's a unicellular organism, only a single cell will take part in all the life processes. So enzymes are secreted into the food vacuole. So then what happens? So the food actually dissolves and the absorption takes place. The food, the energy which has been entered, the absorption into the cell, it takes place. So the next step, actually the food which is digested and the soluble materials are absorbed into the cell, so that some of the life processes can happen. So the utilization of the energy from the food which has been digested for the cellular processes is called as the assimilation. Okay, so the last part, actually after the food is engulfed and all the digestive juices are poured and the food is digested, so then the digested food, the energy is used for the cellular metabolic activities. So the last part is the egestion. The undigested waste materials are thrown out of the amoeba, where the pseudopodia actually moves away from each other, making the food vacuole to just pass out of the body. So this is called as egestion. Okay, so I have some videos for you students, please do watch it.
Here's a cell on your screen. We have already seen that the various organelles inside it are compactly packed inside this delicate membrane called the cell membrane or the plasma membrane. We've even seen that the cell membrane makes use of diffusion process to transport gas and water molecules in and out of the cell. However, diffusion is not the only process employed. There are a few more on the list. Let's have a look at these methods in this video. Tell me one thing, how do we transport water from a lower height to a higher height? We need a pump, because this does not happen on its own. A force or some energy is required to pull the water upwards. Similarly, in a cell, some molecules have to be carried inside or moved outside the cell. This requires energy to be spent. The energy used is in the form of ATP molecules. This is called as active transport. That is, active transport requires expenditure of energy. The transportation is carried out by protein molecules present on the cell membrane. On the other hand, diffusion does not require energy. Hence, it's a type of passive transport. So, movement of molecules without spending energy, as in case of diffusion, is passive transport. Similarly, movement of molecules across the membrane with the expense of energy is active transport. Now let's get to know one more interesting concept named osmosis. Have you ever come across this term osmosis? Well, quite often. But what exactly do we mean by this? Osmosis is defined as the movement of water from higher water concentration to lower water concentration through a semi-permeable membrane. So let's say we have this container which is partitioned by this semi-permeable membrane. Now we add sugar solutions at both these sides. However, the concentration of each is different. Here we add concentrated sugar solution, while here dilute sugar solution. That means the concentration of water in this solution is obviously more compared to this. So after some time, we see water molecules moving from this side of the partition to this side. And how long will this movement take place? It will occur till the concentration of water is balanced, that is, equalized on both the sides. This is nothing but osmosis. Let's learn some more interesting concepts about osmosis and cell transport. We all have seen raisins swelling up when kept in water for some time. Any idea how this magic works? Well, it's simply the process of osmosis which results in the swelling of these raisins. You heard me right. It's osmosis process due to which the water from the surrounding medium enters the cells of the raisins, helping them to swell. Can we define osmosis once? It's simply defined as the process in which water moves from higher water concentration to lower lower water concentration through a semi-permeable membrane. An interesting thing to know about osmosis is that the movement of water is affected by the amount of a substance dissolved in it. And what does this mean exactly? Let's conduct an experiment to understand it better. Let's take 3 plates or containers with salt solutions of different concentration in each. Let's add a few red blood cells, that is RBCs, in each. After a while, we notice that the first set has cells which are shrinked, the second set has cells that are just the same, while the third set has cells which are swollen or bulged. Any idea why this happened? Let's understand this. In the first case, the water content in the surrounding solution was less as compared to the cells placed in it. In other words, the salt content of the solution is more, and therefore this solution is a concentrated one. That is the reason why the water molecules moved from inside the cells to the outside solution, as the concentration of water was higher within the cells. Hence the cells started shrinking. This kind of solution is called as hypertonic solution. That is the solution with higher salt concentration. Now let's have a look at the second case. Here, the level of salt and water seems to be the same on either side of the cell membranes. That is, both within and outside the cell. Therefore, there was a balance in the movement of water. What do we understand by this? It means that the movement of water was the same in either directions. As a result, the size of the cell did not change. It remained the same. Do you know what such a solution is called? It's called as isotonic solution. The two solutions are isotonic when the concentration of water is the same on either sides of the semi-permeable membrane. Now let's focus on this third set. The cells in this case are surrounded by a dilute solution, that is by higher water concentration. The salt dissolved is less and the water content is higher. Therefore, water molecules move inside the cells and hence the cells get swollen. Such a solution is called a hypertonic solution. In other words, solution with a higher water concentration compared to the other. This is how various solutions and their different concentrations help cells to sustain pressure and to survive. Movement of molecules across the plasma membrane is also influenced by the simple process of osmosis. And asked what exactly this is, I'm sure your answer will be a cell.
So students, I have a small little exercises for you. So answer the following question and please submit it to your class teacher so that I can correct and know how good you have understood the topic. Thank you all for joining. Have a great day. Bye.